go ahead and click it.
Okay, Steve, I think you can take it away.
All right. My name is Steve Byers. I'm the chair of Friends of Hackmatack, and I'd like to welcome you to the meeting this evening. Next year, the Friends will be celebrating with the Fish and Wildlife Service the 10th year since the refuge was established in 2012. We're delighted to be able to share with you the program this evening. You can see the agenda. Despite the pandemic, the Friends have been actively engaged in stewardship, land protection, and conservation and outreach. And you'll hear a little bit about that this evening. But before we move on, I just want to thank Cindi Jablonski for hosting the meeting. And at this point, then I'd like to turn the meeting over to Cindi, and we'll get underway with our program, Freshwater Mussels in Chicagoland. Cindi, take it away, please.
Okay, I'm going to stop the share here.
Great. And so thank you, everyone. And thank you for the environmental defenders of McHenry County who are letting us use their Zoom line tonight. I'm going to introduce our first two speakers, and while Laura's bringing her presentation up, and then I'll introduce our third speaker. So starting us off tonight is Laura Barghusen, who's the aquatic ecologist at Openlands. And Laura works with governments, communities, and other stakeholders to connect people to the waterways of Northeastern Illinois through water trail planning, biological monitoring of rivers and streams, education, and events. And Laura is a co-author of a field guide to freshwater mussels of Chicago wilderness. And the next presenter working with Laura is Kentaro Inoue, who is an evolutionary and conservation biologist who joined the Shed Aquarium Center for Conservation and Research in 2019. Kentaro has studied freshwater mussels all over the world. Now he's working in the Great Lakes region to understand how human activities and environmental change affect mussel diversity. So Laura and Ken are going to start us off by telling us about their work on how they use long-term monitoring to save the most endangered critters. So Laura, do you want to try and, oh, there we go.
Okay.
Looks great.
Let me see if I can put it into presentation mode. Okay. All right.
Beautiful.
Very much, Cindi. And thanks everyone for having me here to speak about long-term mussel monitoring in Hackmatack at Hoffman Farm. I'm Laura Barghusen, Blue Ways Director at Openlands. And Openlands is a nonprofit that protects and enhances natural land and water throughout the Chicago region and connects people to nature where they live. Openlands is actively protecting through acquisition and through restoration land in the Hackmatack Wildlife Refuge. And we've partnered with biologist Kentaro Inoue from Shed Aquarium to set up a long-term mussel monitoring site at Hoffman Farm, which is one of the sites in Hackmatack that Openlands owns, and through which North Branch Nippersink Creek flows. As most of you probably already know, mussels are important indicators of stream health. They're sensitive to pollution and they're sensitive to habitat conditions. And long-term monitoring basically means monitoring every year into the future. And long-term monitoring sets us up to get a good sense of the survival and reproduction of mussel populations in the creek over time. And it also opens the door to understanding how any future actions that we take to try to restore or enhance conditions for mussels in the creek are working in terms of how populations are responding. Hoffman Farm is one long-term mussel monitoring site among several that Shed has established in the region. Openlands is really pleased to be collaborating with SHED on this study. And in this photo, you can see Kentaro, who will be talking pretty soon, tagging mussels at Hoffman Farm last summer. Openlands has been working to enhance the understanding of the condition of mussel populations in the greater Chicago region, or in what we call the Chicago Wilderness region for years. And one of our efforts involved partnering with Roger Klosek, who worked at SHED Aquarium at the time, and also Jim Bland, to produce a field guide to the freshwater mussels of Chicago Wilderness. This came out in 2008. And the purpose of the guide was to enhance the ability of conservation agencies and biologists to identify and survey mussels and to help assess stream and river health. We worked with an intern from the Art Institute to create a character named Dr. Clammy, who appears throughout the guide to teach users about mussels. And you can see Dr. Clammy on one side of the screen here. And we work to provide lots of photos to help with mussel identifications. Mussels can be really hard to identify, so we were really trying to provide visuals that would help. And Jim Bland took the photos, and you can see here his photos of umbos at the center of the hinge there. And the shapes and lines of those umbos can be a key trait for identification. And you can also see Dr. Clammy here discussing how dams threaten mussel populations. When native freshwater mussels reproduce, they release larvae called Glaucidia into the water. And these have to attach to the gills of a fish host and travel on the fish until they grow into juveniles and drop off and are ready to live on their own. Each species of mussel has a certain set of fish that they can use as hosts. And some mussels are famous for the lures that they use to attract fish. Some of them have structures on their soft inner tissues that look like little fish or like worms. And they can put these out to attract fish hosts over who are looking for food. And then they release Glaucidia onto them. So this reproduction through fish hosts results in mussels being dependent on the presence of certain fish. And if those fish disappear from a stream system, the mussels dependent on them won't be able to reproduce. Recent research has also shown that mussel Glaucidia, in some cases, can be extremely sensitive to certain pollutants, which also can endanger the mussels. For example, fat mucket larvae are very sensitive to chlorides, which can enter waterways, for example, through road salt. So this map shows the location of Hoffman Farm in the Tamarack core area of Hackmatack. You can see it, I think you can see my cursor here. You can see it as this green square on the easternmost part of this map. And you can see North Branch, Nippersink Creek running through it kind of diagonally. And this is in Richmond, and it's really near Nippersink Library, if that helps place it for you. And this is a picture of the long-term mussel monitoring site that we set up at Hoffman Farm. You can see in it these really skinny orange strings that basically delineate a 15 by 15 meter plot. And it's within that plot that the mussels are monitored each year. The first step in establishing the long-term monitoring site at Hoffman Farm was surveying mussels at sites throughout the creek at all of these locations where you see the push pins on this slide. So this monitoring was led by SHED, and it was done over several days in 2019. The surveys involved searching for mussels and identifying and counting all the mussels that were found at each site. And this is a table where you can see the sites across the top and the mussel species found across the side of the slide. So overall, within the creek, there were 12 species that were found on the Hoffman Farm property. And diversity of species richness at each site varied from, I think, 3 to 8. And after looking at this data, it was decided to create a plot for the long-term monitoring between sites 3 and 4 right here within that red circle area. So, so the outcomes of the 2019 work were that the total of 12 live species were found in the creek on Hoffman Farm, including the black sand shell, which is a state threatened species, and also including the fluted shell, which is a species of greatest conservation need from the Illinois Wildlife Action Plan. So those were all found. And we set up the long-term monitoring site. And each year at the long-term monitoring site, all individuals that can be found within the plot are captured, identified, individually tagged with a tag that's kind of like the microchip that goes into your pet. And Ken will be talking more, I think, about that, and also measured. And you can see that was a mussel being measured last summer at Hoffman Farm. And so over time, recapturing them every year will help track the long-term survival and reproduction of these populations. Genetic samples are also being taken for analysis, and those can deepen our understanding of the diversity within the population and also allow comparison with populations at other sites in the region. So, so we set up the, or we chose the long-term monitoring site in 2019. And so far, we've done monitoring in 2020 and then again in 2021. And these are some photos from the monitoring. You can see Openlands and shed staff and interns working with the mussels, tagging them and recording data on them. And also on the left there, you can see the genetic samples, just a tiny tissue sample being put into vials for further analysis. So basically what we, in terms of the number of mussels that were marked with pit tags in 2020, we caught 96 mussels in the 15 by 15 meter plot, and those were all tagged. And then and put back after being identified and measured and having genetic samples taken. When we came back in 2021, we caught 35 of that 96 that we had found last year. So 35 mussels that we found were tagged by us last year. And then we found another 75 new mussels in the plot who had not been tagged last year. So the total number of mussels that had been, and those were all outfitted with pit tags in 2021. So we had 171 mussels marked by 2021. And the recapture rate between 2020 and 2021 was 36.5%. In 2020, we captured 9 species of mussel. And in 2021, we found 10 because we found a 3 ridge in 2021. And this is just a list of the mussels that we found. in 2021. And you can see again the black sand shell, the Illinois threatened species, and the fluted shell, the species of greatest conservation need. And then as a sort of a surprise, we found out that we had also found an ellipse in the area through genetic analysis. So that's yet another species. And I put also the mottled sculpin, which is one of the ellipse's host fish in this picture too. And this is a fish that is a cold water or cool water species. And this fish, plus some other fish, are the ellipse is dependent on them as host fish. So at this point, I'm going to hand this over to Ken to talk more about the long-term monitoring. But we were very excited to find out that we had also found an ellipse. We have been involved in some work to do surveys to figure out where ellipses are throughout the region and where they have their greatest concentrations. We didn't think that there were ellipses in North Branch Nipperson Creek, so this is pretty exciting. All right, with that, I will stop sharing and hand it over to Ken. Thank you.
Hey, Laura, while Ken's pulling his slides up, maybe you want to start now, just answer one question and then the first question I have is, do the chip tags allow you to relocate the mussels on future surveys?
Yeah, there's actually a device that looks to me like a metal detector. Is that what you call when you look for metal on the beach?
Yep.
Device that looks like that we take, and you can walk through the plot with it, holding it just a little bit above the stream bed. And then I can't remember if it makes noise or if it beeps a light or if it does both, but it lets you know when there's a mussel that's tagged right underneath. And then sometimes it's surprisingly hard to find it, even though it's right there.
Cool. Well, we got lots of more questions in the chat, so we'll let Ken talk about his part of this work, and then we'll keep putting your questions in the chat, and we'll hit up both Laura and Kentaro after Ken's talk. So take it away, Ken.
All right, thank you, Cindy and then Laura. Can you guys hear me? Okay. So Laura gave us good overview of our mussel monitoring effort in Nipperson Creek. So I want to give you a more broad scale, regional scale monitoring effort by shed in the Chicagoland area. So I want to start with, some of you might not know, shed has active research program in the institute. So from marine research to freshwater research. And then I'm a research biologist at Shed Aquarium. I joined the aquarium in 2019 as freshwater mussel biologist. And then since then, I'm working on freshwater mussels, and then I've been working on freshwater mussels for a long time. So kind of overview of what Shed research program and conservation program does. Is I have two main research work. One is field work, and I'm going to talk about this in this presentation. But basically, we are documenting local species richness and diversity distribution in the Chicago area and in Great Lakes in general, and monitoring their health or population health over time. And another project that I'm working on is, Laura kind of mentioned that, is genetic research. We are taking genetic samples from species and populations to understand like genetic diversity and taxonomy and evolution, how those populations are evolved and how they are doing in genetic way, and try to assess genetic hotspot of the population across the region, and how they are, genetically related to each other. And our overall effort is going to be in adaptive management, where we use those findings to make decisions on how to most effectively protect or save those animals and habitat. So I'm going to talk about long-term monitoring, and Laura already mentioned some of them, but the main objective or goal is to try to understand health and stability of freshwater mussel communities in the Chicago region over time. So I have two specific objectives for this project. One is monitor changes in community composition, so how species are changing over time, and also population dynamics, so such as survivorship or recruitment over time in that particular population. We also want to know the effect of environmental factors, such as warming temperature or flood or drought on those mussel survival, and then how they affect to community composition change over time. So for this long-term monitoring project, we have 3 monitoring sites, so including the Hoffman Farm with Openlands. So I have Nipissing Branch, North Branch in Nipissing, and a Hoffman Farm in McHenry County. I have South Branch, Kishwaukee River. I'm working with DeKalb County Forest Preserve District. And then I also have one site in a Poplar Creek in Cook County. So those 3 sites are kind of different landscape and land use, right? So Poplar Creek is more like a residential area. South Branch, Kishwaukee River is more like agricultural field. And then North Branch, Nipissing is a kind of mixture of, logging, more nature area to agricultural area. So we pick those 3 sites to understand how those environmental factors affect mussel communities. So Laura already kind of mentioned that each site we have specific area of the monitoring, and at each site we basically find all mussels from that site, and then tag with a specific number. We call binary tag, which has specific number for each individuals, but also we put it's called pit tag, which is a transponder similar to a microchip in your pet. So basically we can find through this kind of device. It's called pit tag reader. So we can find those animals over time. So I'm just going to talk about the current effort for the long-term monitoring, and then Laura already mentioned some numbers, so it's going to be a little overlap. But we initiated long-term monitoring in 2020. Total, we tagged 644 mussels across 3 sites, so we have 16 species total across Chicago area. So this is kind of breakup of numbers. So Nipissing Creek, Poplar Creek, and then South Branch Kishwaukee River. Numbers are pretty different. Nipissing Creek, we have 96 individual tagged in 2020 compared to South Branch Kishwaukee River. We tag 413 animals. And this year, 2021, we recaptured a total of 493 tagged animals, which is 76.6% recapture rate, which is kind of high. And we also found new animals, so 268 new mussels in each site. So total close to 1,000 mussels we tagged over 2 years. So this is the number of break up by each site. It's interesting that the Nipissing Creek recapture rate is only 36.5% that Laura already mentioned, but the 2 other sites are much higher, more than 80% recapture rate. So most of mussels are actually recaptured from last year. And then also interesting that the total number we found in each year is actually similar or even higher. So actually population size is increasing over time. This is only a 2-year data set, but overall we are seeing increase in population size. So this is a graph breaking up by species. So we are looking at the abundance of each species and across 3 sites, and also 2020 and 2021. So the Nipperson Creek, Market, and then Round Pictos are most abundant animals in that site in both 2020 and 2021. But if you look at the Poplar Creek site, ellipse is actually the highest, most abundant species in that creek. And then Kishiwaki River, we have 12 species overall in the site, which is the highest species richness across 3 sites. And then, but Market is the most abundant animals in that site. And then we are seeing kind of increasing trend of almost all species in the site, which is good news. So I want to kind of break up to kind of significant species in the Chicagoland area. And then the first one is Market. It's most common and abundant animals in 2 sites. And so 38% of all animals we found in the Nipperson Creek Market. And ellipse is also kind of significant species around here because this species is widely distributed, but very limited. They occur very limited locations, but it's most abundant in the Poplar Creek. It's 91.4% of animals we found are ellipse. So it's very abundant there. And Laura kind of mentioned that we actually found 1 freshly dead individuals in Nipperson Creek. This was kind of haphazard sampled. I found 1 small animal's freshly dead, and it looks really like ellipse, but I was kind of doubt. I had some doubt, so I kind of ran the genetic analysis, and the genetic analysis showed this was actually ellipse. So next year, I really want to look into more whether or not we actually have ellipse in that monitoring location. We might be some misidentification with Market there, but so we try to understand, we try to find ellipse in that Nipperson Creek. And the next species is black sand shell, which is a threatened species. It's a rare and then usually found in the larger river systems, but we found in Nipperson Creek, which is great. And then so we are monitoring 1 individuals right now. Fluted shell, Laura mentioned, is widely distributed species, but relatively rare in the northern states. But this species is very abundant in all monitoring sites. And finally, Elucto, which is also a special concern species and widely distributed, but usually you can find small numbers, not abundant. But we tagged 12 individuals across the whole site. So we are monitoring those species and how those species composition change over time. So basically we are gathering baseline information, and we found that high recapture rate in the 2 sites, but not in the Nipperson Creek. We found that the population sites are constant or even increasing. That is good news. And each site has highly variable in species composition and abundance. So this is kind of why we are doing monitoring, to understand this trend is staying in the same direction, or we are seeing some kind of differences. So we want to understand how those species composition and abundance change over time. So we will return to these sites in 2020 for the 3rd year monitoring. And at that point, I should be able to do more analysis so we can assess changes in demography and species composition across those sites, and examine association between demography and environment. So we have temperature logger deployed in each site, but also we are monitoring, we can get river discharge data from USGS river gauge. So we want to try to use those information to associate with demography information. And finally, using those data to evaluate what is the potential conservation actions we can do for each site. Yeah. So this is basically the part of the holistic conservation action and adaptive management project. So we try to advance our understanding of freshwater mussels and apply those findings to better conserve freshwater mussels. And then I want to end with this as thriving mussel populations are creating healthy rivers for everyone because they provide a huge amount of ecosystem services. And then just we want to have freshwater mussels in the system. So with that, I want to say thank you for inviting us for the meeting. And then if you have any question to Laura or I, and then if you have time, if we can answer the question.
Yeah, we do have time. Thank you, Kentaro. So yeah, I think we've got some time for questions for Laura and Kentaro. I'm going to kind of run down the list and then people can jump in. So the next question we had in the chat is, what process is used to obtain genetic samples?
So we are taking small tissue clips from each individual. Sometimes we use a tissue swab. It looks similar to like a cheek swab for your genetic samples. So try to minimize the harm to the animals, and then basically bring back to the lab and then process.
Great. Jillian, you had two questions. I think we'll ask the first one now. Your second question is about raising mussels in captivity, and Cindy's going to be talking about that. So we'll save that one for after her talk. But your first question was, are there any efforts to restore habitat in ways that help mussels? Laura or Kentaro, do you want to take that? I'll take it. I mean, for one thing, removing dams can help mussels. That gives them access to larger reaches of streams. So you all probably have other thoughts on that, Cindy, Laura, or Kentaro.
Sorry, I was muted.
Well, there you go.
Here I am. Yeah, so I know that there have been efforts to reintroduce mussels into stream systems, and efforts to assess like whether or not the host fish, are still present in systems. And that I think there's been some modeling work at Illinois Natural History Survey of what kind of interventions or reintroductions, might work best under certain circumstances. But I actually don't know of in-stream restorations, besides, I guess, dam removals, like you said, Cindy, that, that are known to restore habitat for mussels. I don't know if you are aware, Kentaro.
So one of the issues is like sedimentation and stuff. So that bank stabilization and then those effort is very important for mussel community too.
Actually, when you said that, it reminded me, Openlands with Roger Klosick, and probably also Jim Bland, was doing monitoring in a remeandered section of Poplar Creek, which is a tributary to Hickory Creek, I think, as I recall, which is a tributary to the Lower Des Plaines. And after the creek was remeandered, we found, that juvenile mussels colonized it really quickly, much more quickly than we thought they would. The kind of pioneering species like, trying to remember what was there. I think it was, we did find a slipper shell, or more than one slipper shell there, which at the time were on the threatened species list. I'm not sure if they are anymore. But anyway, so it seemed like that restoration was offering, habitats that the mussels, were colonizing.
Great. Amy's got a question about fish species transporting their larvae, but I know Cindy's going to talk about that, so I'm saving that question for after her talk. We have, I think, a number of questions about recovery rate and recapture rate. So for Maggie, can you explain the significance of the recapture rate? And she says, great presentation.
Yeah, so I guess significance is a high recapture rate. So those mussels actually survive over winter or over 1 year. That's one of significance, right? So those mussels don't usually move much. So they usually stay there for a long time. So we expect to have higher recapture rates. But if flood or drought happens, those animals maybe move away or even die at the site. So basically, we are looking at a stable population by looking at recapture rate, right? So, so the Nipperson Creek has a lower recapture rate. That means maybe that site is particularly, maybe those mussels are moving away from the site due to maybe high flow or something like that. But we actually don't know why Nipperson Creek has lower recapture rates compared to other sites. So that's why we are monitoring long time to understand what's happening. Maybe next year we can find more recaptured animals, or maybe next year it's the same trend. Then maybe just high turnover site. The mussels keep moving, but the population size is exactly the same over time. So it's interesting to look at. So yeah.
Great. I think there's a lot of other questions. People had speculations about why they were low in the Nippersink. So I think you kind of touched on all of those. I'm looking through, there's a question from Lynn. How long can mussels safely stay out of the water for monitoring purposes?
They are actually okay being outside of water for a while. We are not trying to expose to the long time. So we usually use a small bin and then fill the water and then keep mussels in the water. So, but some species are very tolerant to drought. They usually, when water is very low, they usually burrow into the sediment and they try to find a moist area. But some species are more sensitive. So we are not really exposing to, to the air for a long time. It just, yeah.
Got it. Thank you. Then there was a question, what are the predators of mussels?
A lot of things are actually predators. Muskrat, raccoons, they really like to eat mussels. Some fish actually eat mussels, although that's a less common. So freshwater drum actually eat some mussels. What else? Some birds like egrets and other bird can eat smaller mussels, but usually those predators eat like a thinner shell mussels so that they can easily open or easily crack, so that they can eat inside of shell. Like those mucket is very thick shell. They don't really have predators.
Cynthia asked a question. Actually, the Environmental Defenders of McHenry County has been doing a study with some researchers down at U of I in Urbana-Champaign on microplastics. And so we've collected samples that they've analyzed. So both the Nippersink and Kishwaukee have had quite a bit of microplastics. So are you finding microplastics in mussel tissue, or can you comment on the impacts of microplastics on mussels?
I'm not in that field, and I know it's a big issue. So I really want to into that microplastic, but I haven't done any plastic study yet.
Work with you when you're ready to digest.
Well, I see some more questions in the chat, but why don't we go ahead and have Cindi pull up her presentation, and we can hear from her, and then we can go back to some of these questions to all three of our presenters then. So while Cindy's pulling up her presentation, let me do a little intro of her. She's the wildlife ecologist for the McHenry County Conservation District. She worked with the Illinois Natural Areas Inventory Update, evaluating natural areas in the 18 northernmost counties in Illinois before she started working for the district, which was in 2012. And there I know she studies bats and snakes, because she gave this group a great presentation on green snakes a while back, fish and mussels, and there's probably many more critters that she's looking at too. So looking forward to you hearing about Cindy's work too. So take it away, Cindi.
All right, thank you, Cindy. So I'm going to talk about some of the work that the district does to help conserve freshwater mussels. Whenever I do a presentation, I always put the district's mission statement at the bottom. You'll see that's our mission statement. I think it's great, and I think it's a good reminder of what we're here for and what we're doing. I also put this link of this article that the Xerces Society wrote, The Merit of Mussels. Why do we care about them? Why are they so important? In case you want to look at that later. We've been doing organized surveys at the district since 1989. We've done a total of 196 surveys, including this past year. I broke it down in the Fox watershed and the Kishwaukee watershed, the different surveys that we've done for you guys to look at. This is the layout of where we've done surveys. The surveys are in red. The Kishwaukee watershed is the blue, and the Fox watershed is the yellow. We've tried to hit mostly, tried to get surveys in all of our sites. You'll look at Pleasant Valley, the outline for Pleasant Valley in the middle lower portion of the screen has nice streams running through it, but we've tried to do surveys there. The stream is so channelized and so silty, it was impossible. We sank into silt up to our knees. So we haven't been able to do anything in there. Oh, sorry. So prior to 2012, we kind of had random and varied time crawls. We standardized our protocol starting in 2012. We do a 10-man-hour crawl, which sounds like a lot, but you can see we have a lot of people, seasonal staff, and other staff that are very willing to get into the water on a hot July or August day. So it takes us usually less than an hour to do a 10-man-hour crawl. And, we put them in bags and we put them on the shore. We kind of cover them in water so they're not exposed for a long time. We identify them. We identify the dead shells too, and then we put them back as soon as we can where we got them. So for this presentation, I pulled together all our data, which I've never done before. And I wanted to see the total amount for both watersheds, and I pulled out watersheds later. The total amount of live mussels we found and the total number of recent dead mussels. The big winner is the white heel splitter, which we find in both the Kish and the Fox watershed, and then the pimpleback. The listed species on the bottom, purple wartyback, spike, rainbow, we hardly ever find them. I found a spike once, we found a spike once 2 years ago, one. I've never seen a purple wartyback, and I've never seen a live rainbow mussel. The little I put down there, we don't find many of them, but I think they're underrepresented in our samples because they're very small and they're kind of have a niche. They have they're found in siltier areas. We just don't find them very often. And when we do find them, it's just, you know, maybe one per survey, but they are not, they aren't rare. We just don't find them. Now this is the, for both watersheds, the percentage of all, you know, pooled together, the recent dead and the live mussels, the percentage that were found that were recently dead versus live. And you heard in the last presentation, they talked about ellipse a lot. Well, we find more dead ellipse than we do find live ellipse. So they are not common, that's for sure. Not up in our county then anyways. What I found interesting too was the black sand shell, which was just delisted this past spring. We don't find many dead shells of them. I don't know. I don't know why that is, but it was recently delisted. Now I pulled everything out by watershed, and I was really surprised that there are no pimplebacks in Kishwaukee. I hadn't realized that before I pulled all this data together. We see pimpleback mussels so often, I just assumed they were countywide, but they're only found in the Fox watershed. And fat muckets are only found in the Kishwaukee watershed. Black sand shells are only found in the Fox watershed. We worked with DuPage recently, the past few years, and they're working on propagating mussels in DuPage County at their Urban Stream Research Center. And when they come out, they're always, they come out to us because we have some pretty conservative mussels. We're very lucky. We have some conservative mussels that they don't find anywhere else. So when Joe was surveying with us, he'd also look at the pimplebacks and he'd go, wow, look at this, a pimpleback. And I'm like, what? This whole thing? They're everywhere. So now I pulled out the percentage of recent dead by watershed. And again, another concerning number for ellipse in the Fox watershed. 65% of the mussels that we found were recent dead. That's concerning to me. The rest of the stuff wasn't really remarkable. I only put the ones that mussels that were found in both watersheds on this graph. If you want to know what's happening in a stream, you look at a single mussel survey in that stream, and that will tell you what's going on in there. This is our Beck's Woods North site. We did a mussel survey on the Piscasaw in 2016. We did our standard 10 man-hour survey, and we only found 2 live mussels and 4 recently dead and 2 species. And if you look at the area, you think, well, what's the problem? Because it's all buffered. There's some nice natural areas surrounding what's going on there. And then if you zoom out, you can see our mussel survey down there in Beck's Woods. And if you look upstream, you've got all this channelized streams with very little filter strips. Filter strips is the amount of vegetation that buffers the row crop field from the stream. Even if we had that whole site buffered, all that upstream agriculture with no filter strips really took its toll on those mussel communities. So what does a good mussel survey look like? This is a very little short sample reach that we did in Glacial Park. But if you look at what's upstream of it, it's all meandered, it's all protected. And upstream of that is Wonder Lake Dam, and no, dams aren't good for mussels, but in this case, it might hold back some of the siltation that might come down that stream from Wonder Lake. And on that survey, we had 475 live mussels and 37 were under 3 years old. So there's good recruitment going on there. There were so many mussels that we had to stop mid-survey and count them. Our bags were so full. We had to stop and empty the bags, count them, and then record that, and then continue on to finish the survey. And that, again, was our 10 man-hour survey. We've also done a lot of mussel relocations over the years. Most of them are under a bridge for bridge constructions. On the top right-hand screen, you can see Route 31 bridge construction. And you look at that water, and that's pretty standard for under a bridge, very silty, very just nasty looking. But there were still mussels in there, and we take them and we move them up about 200 to 300 feet upstream. The idea being, if we move them upstream, when they do the construction, all that silt and debris won't wash over them if we put them downstream. We also moved some mussels from County Line Bridge construction over on the Boone County, McHenry County border there. And you can see we had a couple divers even, Brad Woodson and John Peters did some scuba diving to get some mussels that were in some of the deep pockets. On the left, we have a whole bunch of volunteers that helped move mussels when we did our dechannelization of Nipperson Creek in 2000. I wasn't here then, but I found this wonderful photo of the community really pitching in to save the mussels before all the heavy equipment came in to re-meander that stream. What I like about this is you got kids in there, you got older folks in there, all these people trying to help out and save these mussels. We were talking, they were talking about, Laura and Ken were talking about tagging mussels. Relatively recently, they've asked us to glitter tag some of our mussels. 50% of them, they want a glitter tag. It's kind of a poor man's system of tagging mussels. Put some super glue on, some glitter, and put them back. I don't know how well it holds up. I personally have never, found one with glitter on it after we put them back in the stream, but that's what we do. I know Illinois Natural History Survey does that too. So we were able to show with mussel data how important filter strips are. Remember, filter strips are that area of vegetation between row crops and a stream that helps with the, keep from erosion and keep from chemical runoff into the stream. In 1954, this aerial photo shows channelized streams with absolutely no filter strips. The district purchased this parcel in 1990. And you can see the healthy, generous filter strips that were put in there. The mussel survey was done in 2005, and no live mussels were found. But then after years of having these filter strips in there, we did a mussel survey in 2012 and found 199 live mussels. They don't just, it takes a while for mussels to repopulate. It just does. So the district now has it written in our agricultural leases that there must be a minimum of 65 feet of filter strips that are maintained on all agricultural fields next to streams. I think an RCS recommends 35 to 36 feet, and that's for highly erodible land. But we have went well beyond that with our 60 feet filter strips. I'm very proud to say. So we worked with DuPage, Urban Stream Research Center. They are propagating mussels in their facility. It's a fairly new science. And as I also mentioned, they come up to our streams because we have some of the more conservative species. In this case, they were looking for fluted shells. And we went out there on a chilly October day in 2017 to retrieve some mussels and to see if they were gravid. Gravid means they're holding larvae in their gills. To make it easier, we had done a mussel survey on that stretch of stream, and I had kind of gathered some fluted shells that we'd found on that and put them in a central location so we could find them. We ended up finding 6 gravid fluted shells. You can see on the right-hand side, Joe from DuPage is just cracking it open a little, to take a peek in there to see if the gills are inflated with Glochidia, which are the larvae. And in the bottom right-hand corner, you can see a gravid female. That's what it looks like when their gills are inflated with Glochidia. When after they transported the gravid females to their facility, they released their larvae, and then they engraved them with this. Each one had a different number. The 17 refers to the year 2017. I can't remember what the P in the 4th stands for. I think the P stands for parent, and not sure. I can't remember what the 4 stands for. But anyways, we put them back right where we found them. And Jesse was very nice. Jesse DiMartini was very nice and sent us these updates periodically. This little video is of 2.5-week-old little mussels. They're 500 microns, which is 1/1,000,000th of a meter, 500 millionths of a meter. And you can see some of the dead shells, if you'll notice the little hooks on them. I know Laura touched upon their unique life cycle. They will release their larvae, and the larvae will go into the gills of the fish, and they will latch on there. You see those hooks there? They will latch on into the gills of the fish, and they will kind of feed off the fish. Doesn't hurt the fish, and then they drop off at some point. She also sent us this little petri dish of 14.5-week-old larvae. That's what they look like under a microscope on the lower left-hand corner, and that's what they look like to the naked eye in that petri dish. I think they had something like 5,000 larvae they counted at one time. And as I mentioned, it's a new science, so there were a lot of challenges they had to deal with. So our fluted shells, we ended up with like 11 that we released into the North Branch, and they were all pit tagged with the same numbers that Aquarium used. Those, not pit tagged, I'm sorry, labeled with those tags. In 2019, we released some creepers. Creepers aren't as conservative. There's more of them, so they just got glitter glue. You remember that girl that we released in 2017 that donated her larvae? We found her again in 2018, and we found her again in 2020. So she's alive. In 2020, she was alive and doing well. So it's good to see that. And here's just another photo of a good survey on Spring Grove Fen, another site that's well buffered. We ended up with quite a few mussels there, and I did leave the Meredith mussels link if you want to look at it later. And that's kind of all I have.
Great, thanks, Cindy. And I think you definitely answered an earlier question about things we can do to help mussels by improving the upland habitat that surrounds the creek and upstream in the creek. So that was great. We got a number of questions here in the chat. Can you determine when a mussel has died by the extent of shell degradation? So question on how,
Well,
you-
we call them recent dead if they aren't real, if the shells aren't real rubbed down, or, you can tell just if they look really old by the wear that they have on them. We, unfortunately, this past summer with the drought, we did a mussel survey where we always had a phenomenal number of fat muckets in South Branch of Kishwaukee, and there were recent dead, as in flesh was still in them, died within the past week or so. You can tell that too.
Oh, thank you. That because Jim Bland had asked that question earlier. Are you seeing influence of stream temperature and drought influences?
Yeah, we called, I called that survey short. I cut it short. I was just so alarmed, and I didn't want to stress the mussels even more by having them out of water at all. So we just cut it in half. It was very alarming.
A question about the average lifespan of black sandshell mussels in regards to their low rate of being found dead. So
yeah, I don't know what that was about. I really don't know why that number was so low. I don't know. I can't speak to that.
A couple of questions asking about can you describe what the district is using in terms of its filter strips? What are you asking your?
Well, they're now 65 feet wide. They're a lot of times they're a mix of some trees are there, some shrubs are still there. We are planting, I think, a mix of native and non-native now. We might be going to completely native vegetation. It's kind of still evolving, our farm program and improving it and our conservation efforts.
Then a number of folks raised concerns about glitter being a microplastic, and
I don't know about that. We don't use a lot of glitter. I'll say that. I don't know. They just started asking us to do that, and I know the history survey does that too. I don't know.
So that's a requirement of when you're.
It was a requirement.
IDOT or?
Yeah. I think that was IDOT, because this was when slipper shell was listed. Slipper shell is delisted along with black sand shell. So we're not getting as many requests to do that with the glitter. We still on our own will relocate mussels. We'll just do that on our own, whether there's listed species there or not.
Then I think there were a couple of earlier questions that any of the three of you want to jump in. There were some questions related to lakes. Don Wilson asks, why do some lakes have mussels and others do not, or at least he hasn't seen them? And yeah, anybody have an answer about mussels in lakes?
We don't survey much in lakes. I don't know.
I know there are certain, mussels that live in lakes and impoundments, even though I think a lot of them don't. But I'm not exactly sure why some lakes would have them and some would not. I'm not sure. I think, do you have any idea, Ken?
My understanding is a lake system usually have less diversity of mussels. Some mussels, only some mussels can tolerate like lotic conditions, so not water flow. So like giant flora or white hill spreader, they are very tolerant to like lotic conditions. So just less diversity than compared to like a lentic system, like a river system. And then I'm sure we don't usually survey lakes and ponds for mussels. So I'm sure we are over, that maybe not looking properly in those lake systems.
Carol had a question say that, they see a lot of zebra mussels in freshwater lakes. So she wanted to know what's the impact of zebra mussels on freshwater mussels in streams.
We never find live zebra mussels in streams. We find some dead shells once in a while. They're just not a stream mussel, right, Ken?
Yeah, my system doesn't really have zebra mussel or quagga mussels, but they basically compete with the food resources because they are also filter feeders, and then they reproduce quicker than freshwater mussels. They produce large biomass compared to freshwater mussels, and then they die pretty quickly. So that actually kind of degrade habitat for freshwater mussels. Not around here, but in the south, I've seen some zebra mussels in the stream system. I don't know how much impact actually on the freshwater mussel system, but Great Lakes, we have so many quagga mussels and pretty much extirpated native freshwater mussels.
Cindy, can I respond to that?
I was thinking that we had a lakes expert on the call here, so go ahead, Jim.
Yeah, no, we do see lots and lots of zebra mussels, and the zebra mussels are directly downstream from lakes. In Lake County, you have 30 plus lakes, even more than that. There's 190 lakes in Lake County, and the number of lakes that are infested with zebra mussels is substantial. We do see them downstream from the lakes. They do survive over a period of time based on their size that we see. And then this past year, the business of the drought, we're seeing large sections of stream that have dried up completely. And very, very unique drought conditions by comparison with previous to it. But the bottom line is the zebra mussels are widespread in regional lakes.
Thanks, Jim. I think we got a couple of last questions here. A question about engaging young people with mussel education, high school environmental clubs. Any of you working on outreach to schools about mussels?
Openlands has not really done that, but I am pretty sure Friends of the Chicago River is doing that for the Chicago River. Doing education programs and surveys with, at least, not sure if it's schools, but volunteers and groups.
Okay, thanks. Okay, kind of our last question is, what do you all think of this reintroducing mussels that have been raised in captivity, like the ones at the DuPage facility? Do you really feel that offers promise for?
I think it's wonderful. I hope they get really good at it, and I hope they are very successful with it. I don't, but until we protect those streams, it doesn't matter how many, mussels we put back into them until they're protected.
Thanks.
So I'm
ahead.
actually working on kind of different perspective of propagation. So I'm also working with Jesse DeMartini at the DuPage County. So I wanted to look at the genetic impact by propagated juveniles. So we actually take genetic samples from offspring from single broodstocks, so a single female, and how much genetic diversity are sustaining within a single broodstock. And then we found out it's a good amount of genetic diversity, but also introducing those large number of propagated animals from few females may change genetic composition in a local population. That may not be good idea for long term, just because we are artificially changing genetic diversity and genetic structure, which may be evolved or adapted to the local environment. So I'm a little bit cautious about propagating and then introducing large amount of individuals into local population, just because we may be impacting and introducing some artificial selection into the system. So that's one thing to think about.
Well, lots of things to think about, and I'm sure you all are going to be learning more as you continue your work and your research. So we really appreciate hearing from all three of you. You guys can use your reaction buttons to do virtual applause. So thank you, Laura. Thank you, Kentaro. Thank you, Cindi, for a great set of presentations. Oh, we're seeing lots of clapping going on. So thank you all so much. I'm going to turn the program back over to Steve and for the rest of our program this evening. And be sure our presenters be sure to be looking at the chat, because there's lots of kudos coming there too. So
Cindy, you want me to do the screen share?
Yeah, put it up.
yeah, or I can do it now if you want.
I've got it.
You got it? Perfect.
All right. Well, I would like to echo the thanks that Cindi just provided for our three speakers. Thank you, each and every one of you. And I'm sure that we're well appreciated by our people and our attendees. As you can see from the agenda, we have a few things that we'd like to just touch bases with. And I've asked Ed Collins, our vice chair, to provide a land protection update for the refuge that's now 9 years old.
Thanks, Steve. I will be happy to do that. What I can say to you is that since the refuge was authorized in 2012 up to today, there's been a great deal of activity in terms of land protection efforts. And those efforts have been completed by a really unique partnership called the Hackmatack Land Protection Partnership, which right now contains, I believe, 11 different groups that are working actively in some aspect of land preservation within the Hackmatack refuge area and the areas right around it. So I could tell you as of this past July, there had been 20 different land protection projects that had preserved 1,569 acres within the refuge footprint. And just outside the refuge boundaries in what we call the greater Hackmatack ecosystem, which I'll talk briefly about in a minute, there were an additional 6 projects that had preserved 474 acres. So when you total those together, since ecologically the plants and animals don't look at the boundaries too closely, that was over 2,000 acres that have been protected in Illinois and Wisconsin since the refuge came into existence. Now, some of that land has been transferred to the refuge proper and is now actually part of the refuge itself. Other parcels are awaiting that as funding becomes available. And some remain in private hands or in the possession of some of the partners. I'll also give you an update. Since July, there have been 2 additional projects that have preserved another 256 acres. And I'll just talk briefly about those 2 projects. One has been within the one of the Hackmatack core areas in the Hebron peatlands, which is close to the Wisconsin border, actually very, very close to it, right on the border. And that particular project preserved a 200-acre tract that was mostly wetlands, that contains some wet mesic prairie remnants. But I think key to what we've been speaking about tonight, it contains a long stretch of the headwaters of the north branch of Nipperson Creek. So that's good news for the mussels that we have been talking about tonight. Just outside the refuge boundary, there's been an additional project completed this summer at the Lake Elizabeth Conservation Area, which lies literally just outside the boundary, with the help of a truly conservation-minded family that had been investigating ways to preserve their family farm for a number of years. It was purchased by the IDNR, which is one of our partner organizations in the Hackmatack Land Protection Partnership. And it protects a piece of that high quality lake, some substantial mature oak groves, and most importantly, some archaeological resources that we know are in that area. To kind of sum it up, there is also another project currently in the works, which is not yet completed. So I'll be a little bit vague on where it's at, but it contains a very large wetland complex, the last piece of one, which will allow some additional hydrologic restoration to be undertaken once it's been protected. It too has very nice, large, mature fir oaks on it, and it protects a portion of a high quality Illinois natural area inventory site. Lots of activity since 2012, more to come in the years to follow. And I think what it really says is that refuges are built 1 acre at a time. In 2000, when Hackmatack began, it was just an idea. When it was authorized, it was an idea that had form but no acreage. And now we approach 9 years later and we see over 2,000 acres of land that has been permanently protected in some form or another. So conservation is an iterative process that occurs generationally and over time. One thing I'll close with, the Hackmatack Land Protection Partners are currently working on defining what exactly that greater Hackmatack ecosystem means in terms of boundaries, but it's certainly an area that is larger than the refuge, but contains lands and waters that are ecologically interconnected to the refuge, and includes important natural heritage elements of the region. So that tells you where we've been the last 9 years and where we've been the last couple of months. And hopefully next time we meet, we'll have some new projects to talk about.
You need to unmute me.
Thanks, Steve.
Okay.
You are.
All right, thank you, Ed. Conservation outreach, Nancy Williamson is going to highlight some of our outreach activities over the course of the last year.
Okay, well, as we all know, this has been a challenging journey for all of us. Luckily, we're in the business where we can do things outside. And so we have been able to hold some very successful friends gatherings outside to show some of our work for the last year and a half, basically, under the COVID strain. So we have held 2 friends gatherings this year, and a virtual meeting about orchids, which was native orchids, which was very well attended. And then a field trip to some of our great conservation partners, June and Steve Keebler, in their parcel near Alden. And we had great attendance, even though we have to register people and limit the amount of people. Everybody is very interested in seeing what's happening. We had a small window where we actually were able to this year hold our Monarch Coalition events, our Monarch Family Fun Fair, with multiple partners. We do not do this alone, but we focus on the drive from Fish and Wildlife to make sure that we maintain monarch and enhance monarch populations and pollinators. And we hold a big event in Crystal Lake at the main beach. And this year, once again, we had an overwhelming number of people. More than 500 people attended the event. We had more tents outside, but we still had tents inside, and lots of people who were willing to take masks and do the right thing. And we had a great time with that event. So everybody is very much willing to come to our Eagle Days, to our migration days, and they want to know about these things. So it's very heartening.
Thank you, Nancy. So there's been a lot of outreach activities, reaching out to a number of individuals. I've also asked Nancy to just comment briefly on the Stewardship Challenge Grant with the Friends and the Illinois Clean Energy Foundation.
Okay, once again, we've taken on a parcel with Openlands. Ed talked about parcels that will be turned over, and Openlands is a terrific partner. And hopefully we will get this parcel into shape. It's right near Richmond High School, on the northern border of Richmond High School. And to just give you a brief thanks to our multiple members and partners and people who donated, we were able to raise $7,000, and Illinois Clean Energy Community Foundation gave us $21,000 in cash. And then we were able to log well over 400 hours in volunteer hours. If you hit 400 hours, they give you $4,000. And then finally, between our volunteer board members, we logged 200 hours in social media and advertising this. And so the bottom line is we're able to secure $34,000, all of which goes to restoring that parcel, which we call Tamarack East. We had high school students coming out. We had first couple of events, we had so many people, we had to have registration because of the limit to making sure we followed the COVID protocols. But we have people there all the time working, and it's been a terrific show of the community, thanks to some very hardworking board members, including Pete Jackson and Steve Byers. And so we're almost done with that grant. It's approximately a 2-year grant, and we've seen a huge transformation in that parcel.
Okay, so thank you, Nancy. Todd, you're up with Refuge Manager comments that you'd like to share with us and our friends.
Yeah, I can add a few things in there. So I'm always impressed with what the Friends group does and how much they really contribute and help out the refuge. And it doesn't go without notice. So to circle back kind of what Ed talked about, this year, actually, the refuge received $500,000 from the Land and Water Conservation Fund to be used for acquisition. And then there is also an additional $1.2 million received for My Bird money. So the My Bird Commission went in front of the My Bird Commission out in DC. The project was actually approved, which is a very significant thing for the refuge because it was actually the first time we've ever done it. And because they approved it, the next time is a lot easier. So just for this year, we received about $1.7 million for land acquisitions. So it did get noticed of all the great work that's being done in this area. The other thing was for the community outreach or the conservation outreach programs. This year I was able to hire essentially an intern through the Hispanic Access Foundation. They allowed us to essentially hire on a Hispanic individual to help with outreach efforts. This particular individual, Sunny Meva, was able to help us to reach out more to the Latino communities to try to really get them engaged within these outreach activities. But he ended up being a great hand overall with all the activities. He was willing to sit at the tables and talk to people and really start to mix up with people. And then the last thing I'll touch on is congratulations to the Friends Group. They did receive the Invader Crusader Award, which is an award that was given by Wisconsin Invasive Species Council for all the invasive work, removal work on garlic mustards, primarily the species on the Turner parcel. So this year for 2021, they received the Invader Crusader Award. And that's all I got. So I'll keep it short and sweet.
Thank you, Todd, and thank you for your leadership in reaching out and helping build the refuge. You see under the next agenda item, member vote on board, and we have 2 items that we'd like our members to approve. The first is a 3-year extension for Vince Mosca and a 3-year extension for Paul Brewitt. And I would entertain a motion from one of our members to approve the action to extend 3-year terms for both of those individuals.
I'll move that.
And who moved that?
That's Tom.
Okay, Tom, thank you.
And
it's
I
been-
second that.
and it's been seconded. And all of those, let's do it by voice vote. All of those in favor, please signify by saying aye.
Aye.
Okay, anyone oppose that action? Well, I'd like to thank Vince and Paul. Vince never ceases to amaze me. He's always willing to go give a talk with the margarita in hand. And Paul Brewitt is our manager extraordinaire, helping out with any number of activities on properties owned by our land protection partners, removing field drain tiles, planting prairie seed, and the list goes on. So I warmly welcome those 3 individuals back on the board for 3-year terms. The next item I have is I'm delighted to share with everyone that a recommendation from our nominating committee to ask Linda Offling to serve a 3-year term on the board of Friends of Hackmatack. Linda's attended many of our outreach activities, many of our stewardship activities, and she brings a number of skills that will be helpful to the group. Design and web skills, organizational skills, and computer savvy. And she has indicated in her application that she thinks we're all a lot of fun. So I'll take that a little bit with a grain of salt, but I do appreciate, Linda, your first of all, your interest and enthusiasm and your willingness to be a part of Friends of Hackmatack. And we're delighted to have you with us. And now I need to submit that Linda's name to the membership for approval on a 3-year term. Do I hear a motion to do that?
So
Yeah, I'll
moved.
move that.
Okay, it's been moved by, was that Ed?
Yes.
And seconded by Tom?
You bet.
Okay, all those in favor, please signify by saying aye.
Or wave your hand.
Aye.
Aye.
Congratulations, Linda, and welcome aboard, Friends of Hackmatack. Would you like to say a few words at all?
Well, thank you. And I just hope I can be of some help. So thank you.
Well, thank you for your interest and enthusiasm. Now I'm going to ask, you see there the Hackmatack Hero Award. In the past, the Friends have presented organizations with the Tamarack Award, but we have not presented an award in the last 2 years. But there are individuals that have made significant contributions to the work of the Friends.
Water? Okay.
So I would like to ask Pete Jackson to present the Hackmatack Hero Award.
Thank you, Steve. And Tom Von Geldern, don't go anywhere. Tom Von Geldern is a
Oh, you
former.
gotta be kidding.
Stay here, Tom. Don't hang up on us. Tom Von Geldern is a former Friends of Hackmatack board member and has a distinguished career as a scientist. But at this time, we wish to recognize Tom for his relentless work as a Friends of Hackmatack volunteer. We are blessed to have outstanding volunteers all around, and extraordinary conservation volunteerism is so common with our volunteers that it's very difficult for any one of them to stand out. But Tom is one such person. He never misses a work day. He comes early, he stays late, and after everyone else has gone home, Tom comes back, often several times, to make sure the brush piles have burned out safely, much to the glee of the Richmond Fire Department. A few of us often do work between work days, and when we need some assistance, Tom is always available. I often tease Tom that he seems to be in a contest with Steve Byers to see who can get the dirtiest. We are so grateful to have Tom's steady and experienced hand with our ecological restoration work, and it's my honor to award the first ever Hackmatack Hero Award to Tom for his countless hours of volunteering in our restoration work and support of the Friends. Tom has put lift into our flight. Tom, thank you so much and congratulations.
Here, here. Well done, Tom.
Well done. Very nice.
Well
Thank you,
deserved.
Tom.
Well, thanks, and that's super embarrassing since you guys are both there before I show up and there after I leave. So I'm not sure where the award should be.
And in addition to the certificate that we have in front of you, we will also be providing Tom with a photograph. Go ahead.
Oh, you know what, I'm gonna have to stop sharing. I don't think we have that on here.
Yeah.
Oh, yeah, we do. Okay, thanks, Cindy.
Ooh,
So we're
wow,
going to.
that is gorgeous.
This is one of Ray Mathis' photos of the Nipissing Creek, and I wouldn't be surprised if there are a number of mussels right there in that stretch.
Exactly.
But Tom will be presenting this image to you as suitable for framing and to put on your wall, okay?
It's framed.
Wow. Well, it's beautiful, and thanks a lot. And boy, it's a great organization to work with. So I just appreciate you guys letting me play along with you.
Thanks for it all, Tom.
Thank you, Tom. That wraps up our annual meeting of Friends of Hackmatack National Wildlife Refuge. Thank you again for the great presentations by Laura and Kent and Cindy. I hope you all have a better appreciation for mussels and what they need to survive. And certainly there are a number of them that are worthy of further effort to better understand and protect. Also, just let me take this opportunity again to wish each one of you the very best for the future, and urge you to continue to be safe, and thank you again for joining us for our annual meeting this evening. So with that, I would entertain a motion to adjourn the meeting.
I'll motion.
Moved by Linda, and somebody seconded that. Who seconded that?
That'll be me, Pete.
All right. All right, that concludes our meeting for the evening. Thank you again, speakers, and thank you all for attending our annual meeting. And hopefully next year we'll be live and in person.
And thank you for your support.
Thank you.
Thank you, Steve.
Thanks for the
Thanks,
organizing,
board
guys.
members, for all the work you guys do.
And Cindy for arranging the Zoom. Nice job.
Mhm.
Yeah, the pressure's on her, that's for sure. Nice work,
Thank
Cindy.
you all.